Assalamu alaikum wa rahmatullahi wa barakatuh. Our research is talking about resistance modulation action of natural propolis against methicillin resistant Staphylococcus aureus. About the past history of Staphylococcus aureus pathogen in human infections, it was commonly found in mucous membranes and the skin of around a third of the population, and it was one of the earlier bacteria in which methicillin resistance was found in 1947. Methicillin resistant Staphylococcus aureus was first detected in Britain in 1961 and is now quite common in all populations. MRSA causes life-threatening bloodstream and surgical site infections as well as pneumonia. WHO first published its priority list of antibiotic resistance in 2017, and MRSA was classified as a high priority, and according to the CDC Antimicrobial Resistance Report 2019. There was a published data describing methicillin resistance in Staphylococcus aureus here in Egypt at 2017, conducting that 40 to 70% of infections caused by Staphylococcus aureus strains are due to methicillin resistance ones. About 24% in Minya City among hospitalized patients there, and from 50 to 82% in Cairo and Alexandria hospitals. And prevalence of community-acquired MRSA in this reported published data was 19 to 47%, and awareness of MRSA control measures among the medical staff at one of the major health centers in Egypt was at 60-70% level. MRSA as a superbug becomes a serious threat to global health and is associated with significant morbidity and hospital mortality worldwide, and has become resistant to almost all available antibiotics except linezolid and dicoplanin, and the intrinsic methicillin resistance of Staphylococcus aureus is due to the incorporation of a MECI gene into its bacterial DNA. Now we must get a solution to overcome the rapid resistance evolution. The emergence of these multi-drug resistant bacteria increases the need for new antimicrobial agents. One of the approaches towards combating drug resistance is to use natural products in combination with antibiotics in a trial to restore the activity of them. Propolis, as a natural product, provides a solution to overcome rapid resistance, and it is one of the most potent natural antimicrobial products from the beehive, and it's considered as a beehive glue produced by honeybees. It contains a complex chemical structure of phenolic compounds and flavonoids, and the antimicrobial activity of propolis are related to the synergistic effect among its components. The major composition of propolis is from 30% wax and 50% resins, 10% essential oils, 5% pollen, and 5% organic compounds. Our aim of work here is isolation and identification of Staphylococcus aureus clinical isolates from different clinical samples, investigation of the antimicrobial activity of phenolic extract of Egyptian propolis, assessment of resistance modulatory effect of ethanolic extract of propolis on MRSA and its synergism with antibiotics in a trial to resensitize them again towards their pathogenic targets. And the methods here conducted in this research was isolation and biochemical identification of Staphylococcus aureus clinical isolates, detection of methicillin resistance between Staphylococcus aureus isolates, C16 disk and BCR for MECA gene, antibiogram of isolated Staphylococcus aureus isolates, ethanol extraction of Egyptian propolis. MRSA as a superbug becomes a serious threat to global health and is associated with significant morbidity and hospital mortality worldwide, and has become resistant to almost all available antibiotics except linezolid and dicoplanin, and the intrinsic methicillin resistance of Staphylococcus aureus is due to the incorporation of a MECI gene into its bacterial DNA. And now we will talk about the results of our study. 130 isolates from 312 clinical specimens were collected from hospitalized patients in Pennsylvania University Hospital, were identified as gram-positive cocci, mannitol fermenter when grown on mannitol salt agar, catalase positive, coagulase positive, DNase positive, and therefore they were confirmed as Staphylococcus aureus and distributed here as shown in the next figure. figure here shows Staphylococcus aureus isolation percentage from different clinical specimens, as 40% was from blood culture, 30% from urine culture, 16% from pus culture, and 10% from sputum culture, and finally 4% from pleural effusion culture. And after isolation and identification of Staphylococcus aureus isolates, we detect the methicillin resistance among these isolates by two methods, the cefoxitin screening test and MIC-A gene PCR. At the right here, this is the cefoxitin screening test, and as shown here, the cefoxitin disc at the center of this plate and growth of Staphylococcus aureus all over the plate, and this means resistance to this antibiotic. And as there is no zone of inhibition around disc as shown here. And at the left, it is the picture of gel electrophoresis of the PCR product here, and the lightened bands here means the presence of the MIC-A gene at 966 base pair. Absence of these light bands means there is a negative result, meaning there is no MIC-A gene in this isolate. This table summarizes the results from the cefoxitin screening test and PCR for MIC-A gene detection. 103 Staphylococcus aureus all were positive for the presence of MIC-A gene, and the cefoxitin screening test result as the zone of inhibition around the disc was equal to or less than 22 millimeters, and according to the CLSI standards, it's all classified as MRSA, while 27 of Staphylococcus aureus isolates were incorporate or not incorporate the MIC-A gene, as 19 out of 27 Staphylococcus aureus isolates incorporate the MIC-A gene, but it is not expressed as the zone of inhibition of the growth around the cefoxitin disc used was equal to or more than 23 millimeters and classified as MSSA. It is methicillin sensitive Staphylococcus aureus. Two standard strains were used here. It is the MRSA ATCC, and the zone of inhibition was 21 millimeter, and of course positive for the MIC-A gene, and MSSA ATCC, it is a sensitive standard strain. The zone of inhibition was 38 millimeters, and of course negative for the presence of MIC-A gene. We will talk about the antibiogram of Staphylococcus aureus isolate. We will start by the resistance pattern of MRSA. All tested MRSA isolates were resistant to ampicillin, penicillin G, and ceftazidime, while resistance to cefepime, gentamicin, clindamycin, and ciprofloxacin shows different percentages. In fact, amikacin and linezolid resistance was the least here, while amikacin shows intermediate resistance among these MRSA isolates, as 6% of them were intermediate resistant to amikacin. And thank God, linezolid completely was used as a last resort antibiotic here, and all MRSA isolates showed sensitivity completely to the linezolid. And as shown here, this is antibiogram of methicillin susceptible Staphylococcus aureus, and we mentioned before that 27 isolates out of 130 were sensitive to cefoxitin. Methicillin susceptible Staphylococcus aureus isolates shown here are all sensitive to ciprofloxacin, amikacin, and linezolid, while shows a high pattern of resistance towards ampicillin, penicillin G. Ceftazidime and cefepime resistance were low here, but the intermediate pattern was more than the sensitivity pattern in case of ceftazidime, while cefepime sensitivity and the intermediate pattern was approximately equal to each other, while gentamicin resistance was very low, in fact, and sensitivity percentage was very high. And come back to Propolis, showing here the methodology of ethanol extraction. of propolis. Sample of propolis used in this research was collected from the beehives located in Kafr Sheikh, Nile Delta region in Egypt, and supplied from the National Agriculture Center here in Egypt. And the propolis sample was grounded mechanically first, then macerated in 70% of ethanol in opaque and stoppered glass bottle. Of course, stoppered to bypass evaporation of ethanol with occasional shaking for 2 weeks in dark room. And then this bottle was put in the refrigerator at 4 centigrade and 2 with occasional shaking to allow all insoluble particles to settle down at the bottom. Then the stoppered cover was removed and allowed the ethanol to evaporate at room temperature till complete dryness. And the result is a sticky, resinous, brownish matter. This is the propolis sample, and in fact it's very sticky and very difficult to handle with it. So we put it at freezer for a few hours for complete freezing, and then we smashed it into powder, then waited, and a working solution was made from this by ethanol. And now we will show the results of antibacterial activity assay of Egyptian propolis against Staphylococcus aureus isolates. Determination of the minimal antibiotic concentration and the minimum bactericidal concentration of Egyptian ethanol extract of propolis. As shown here at the left, there are 4 different patterns of the minimal inhibitory concentrations of propolis. The major one is the minimal inhibitory concentration equal to 0.78. It represents 30.1% of total tested isolates. And at the right here is the minimal bactericidal concentration of ethanol extract of propolis towards the MRSA isolates, and the most predominant minimal bactericidal concentration is 1.56 milligram per milli, and it represents 56.3% of the total tested isolates. Propolis showed 3 different minimal inhibitory concentrations toward this methicillin susceptible Staphylococcus aureus isolates. The most predominant minimal inhibitory concentration was equal to 0.39 milligram per milli, represented 40.74% of all tested isolates, and the 2 other minimal inhibitory concentrations of propolis were 0.195 milligram per milli and 0.781 milligram per milli. While minimal bactericidal concentrations of propolis toward this methicillin susceptible Staphylococcus aureus isolates showed 2 different patterns. In fact, the most predominant minimal bactericidal concentration of propolis was 0.78 milligram per milli, represented 55.5% of all tested methicillin susceptible Staphylococcus aureus isolates. We will talk about the antibiogram of Staphylococcus aureus isolate. We will start by the resistance pattern of MRSA. All tested MRSA and here the time kill assay results of ethanol extract of propolis towards a standard strain used here is a methicillin susceptible Staphylococcus aureus ATCC treated with 2x MIC and 4x MIC of ethanol extract of propolis. As shown here in this figure, the blue line represents the control, and it is a fixed count all over the time of incubation, and as it is not treated at all with propolis. The red line represents the 2x MIC concentration of propolis treated in the standard strain used here, and the viable count shows fixed count from 0 to 4 hours of incubation. After 4 hours of incubation till 6 hours of incubation, there is a bactericidal activity showed here, the decline in the red color here, and the gray color represented the culture of standard strain treated with 4 MIC concentration of ethanol extract of propolis, and showed a bacteriostatic effect from 0 to 4 hours of incubation, meaning a small decline in the count. but after 4 hours till 6 hours of incubation, there is a sharp decline of the viable count. After 6 hours of incubation, there is no culture recovered from the plate. The tested culture is the standard strain resistant to methicillin is MRSA ATCC, and here treated the same way with 2 different concentrations of ethanoic extract of propolis, 2-XMIC and 4-XMIC. And in fact, the red color here represented the 2-MIC concentration of propolis, showed only a static effect on the viable count from 0 to 6 hours of incubation, and no bactericidal activity showed here. And the gray color represents the 4-XMIC concentration of ethanoic extract of propolis treated in MRSA ATCC, and shows a static effect from 0 to 2 hours of incubation. And the bactericidal activity start to be here after 2 hours of incubation until 6 hours. And the big decline here is from 4 to 6 hours of incubation, and no culture was recovered in plates after 6 hours of incubation. After the previous results confirmed the antibacterial activity of ethanoic extract of propolis toward the Staphylococcus aureus clinical isolates, we conducted the study of antibiotic resistance modulation of propolis. The resistance modulation study involves the determination of MIC of standard antimicrobials in presence of sub-inhibitory concentrations of propolis and the minimal inhibitory concentration of the standard antimicrobials alone. The modulation factor more than 2 was set as a cutoff for the biologically significant resistance modulation. Natural extract act as a resistance modulator when this modulation factor is more than 2. The modulator factor is the ratio of the minimal inhibitory concentration of antibiotic alone divided by the minimal inhibitory concentration when it's combined with the natural extract, and here in case of our study the ethanoic extract of propolis. And here in these charts represents the result of resistance modulation study of Egyptian propolis ethanoic extract, of course, towards a multi-drug resistant clinical isolates. The 6 tested antibiotics used in this study were clindamycin, gentamicin, ciprofloxacin, vancomycin, amikacin, and linezolid. In the right chart here, red in color, represented the MIC of clindamycin alone and in combination with ethanoic extract of propolis. Here, the MIC of clindamycin alone was 1024 microgram per milli, and when treated with propolis, there is a sharp decrease in its MIC till 4 microgram per milli, and gentamicin shows a MIC alone equal to 64 microgram per milli, and when treated with ethanoic extract of propolis, there was a sharp decrease in its MIC till 1 microgram per milli. The same as ciprofloxacin shows a high MIC concentration alone equal to 16, and there is a sharp decrease in its MIC when treated with the sub-inhibitory concentration of propolis equal to 0.25. And come back to propolis, showing here the methodology of ethanolic extraction of propolis. Sample of propolis used in this research was collected from the beehives located in Kafr Sheikh, Nile Delta region in Egypt, and supplied from the National Agriculture Center here in Egypt. And the propolis sample was grounded mechanically first, then macerated in 70% of ethanol in an opaque and stoppered glass bottle. Of course, stoppered to bypass evaporation of ethanol with occasional shaking for 2 weeks in dark room. And then this bottle was put in the refrigerator at 4 centigrade and 2 with occasional shaking to allow all insoluble particles to settle down at the bottom. Then the stoppered cover was removed. And in case of tested standard strain, penicillin-resistant Staphylococcus aureus (ATCC), the clindamycin shows MIC minimal inhibitory concentration equal to 32 microgram per milli, as there is a sharp decrease till 1 microgram per milli when treated with propolis. The same as gentamicin, there is a sharp decrease of its MIC alone from 64 microgram per milli till 1 microgram per milli MIC when treated with propolis. Ciprofloxacin showed a moderate decrease in its MIC from 1 microgram per milli to 0.25 microgram per milli when treated with propolis extract. Here, the rest tested antibiotics were the MRSA standard strain ATCC. Amikacin shows a decrease from its MIC test when tested alone from 32 microgram per milli till 4 microgram per milli when treated with propolis. The same as linezolid, there is a decrease in its MIC when tested alone from 1 microgram till 0.0312 microgram per milli when treated with propolis, and the same was with vancomycin, with a promising resistance modulation effect. When vancomycin tested alone showed minimal inhibitor concentration equal to 8 microgram per milli, but when treated with the propolis extract, there is a sharp decrease in its MIC till 0.0019 microgram per milli. From the previous results, we conclude that ethanol extract of propolis, of Egyptian propolis, shows a resistance modulation effect and act as a resistance modulator of these tested 6 antibiotics against Staphylococcus aureus clinical isolates and the standard strains too. Conclusion of all of our study shows that propolis is a promising antibacterial pea product and revealed that the Egyptian propolis exhibit considerable anti-staphylococcal activity and the marked synergistic effect and resistant modulatory action with commonly used anti-staphylococcal antibiotics. Propolis might be used in the development of alternative products for therapy of MRSA infections. Thanks all for watching. Best greetings. Thank you.